All right. First speaker, the second part of the first session is Susan Fisher from UCSF. You can see her bio in the booklet you have. Title of her talk made me a little squeamish, but that's my personal genetics. And I've known Susan for a while and very much looking forward to her presentation.
So I wanted to thank Larry for inviting me. It's always a thrill to be able to combine the two things that my lab does, which is human reproductive biology and proteomics. And so I think you'll get a taste of both. I'm going to give a science-y talk, but I'm going to show you some data. As a scientist, I'm incapable of giving a talk without data. But I also wanted to take you just through the highlights. I also have a very loud voice, and am I speaking so loudly that, is it okay? Okay, good. So I want to start out with this Japanese woodcut because I would like to have you think about something that I think very few people have ever thought of, and that we essentially have nearly a total absence of prenatal care in the U.S. If a baby is born and has a problem, millions of dollars are spent, but before that baby is born, there's no prenatal care. So I want to convince you that pregnancy is the ideal time to start personalized care. Well, why is that? Well, in pregnancy, we have this awesome organ inserted into the mother's body, and that's the placenta. So we have these organs that we're trying to diagnose that have problems. They've been with us our whole lifetime, and we don't know how they change. But in pregnancy, we get an entirely new organ inserted into the mother's blood. So I want to talk about the placenta. Here it is. I hope it's not too squeamish. Is everybody okay with this? Here, okay. So here's the placenta. Here's the baby tethered to the placenta by the umbilical cord. The placenta is really remarkable because it's a fetal organ. It does not come from the mother. And this fetal organ is especially unique because it has enormous cancer-like properties. It attaches to the uterine wall at implantation, but then it invades the uterus so that in pregnancy, a woman's uterus is mostly lined by cells of the baby. So what I've shown is this interplay of maternal and fetal cells in this red box. And this is a real tissue section from cases that were collected in the UK from traffic victims that were pregnant women. And you can see in this section that the placenta, which is the filmy tissue at the top and the uterus, the purple at the bottom, there's a joining of this tissue in human pregnancy. There is absolutely nothing like this that happens in human biology, and it doesn't happen in animals. This is human biology. So this is a diagram that we published to try to explain this, because this anatomical relationship is not something that I think most people appreciate. I speak all the time at OBGYN meetings, and afterwards the OBGYNs come up to me, usually in droves, and say, does this really happen? So I think you can appreciate this is something that people really don't understand. So we have cells of the baby. Here's the placenta, cells of the baby. Here's the uterus, and these cells are invading the entire maternal wall of the uterus. But even more remarkably, these cells take over the mother's blood circulation to the uterus, so that when a woman is pregnant, the cells of her arteries in that organ, the uterus, are no longer lined by her own cells. They're lined by cells of the placenta, which is amazing. And this whole process diverts blood flow to the placenta. So now we have blood that used to go along the surface of the uterus back to the mother, and it's going through the placenta. And the placenta is this amazing structure. It has all these tree-like processes, and it's all designed to exchange with the mother. In fact, it's the only place in your body where you have branched microvilli, so it's just covered with thousands of tiny little projections for transport. And here are some of those tiny little projections. So we think of it in pregnancy and in reproductive biology as a transport organ, but let's think of this a different way. Let's think of this as a source of biomarkers for pregnancy. This is an enormous exchange organ, a new organ inserted into the mother's blood for 9 months. So this is, in my way of thinking, an unbelievable chance to find biomarkers and to transform prenatal care. So let's see what these cells look like. There are a subset of cells. This is all a technique called scanning electron microscopy, which I think is quite beautiful. It gives us this wonderful mirror and window into biology. So some of the cells of the placenta are not transport fuzzy cells like I showed you before. Some of them are invasive cancer-like cells, and that's the cells I'm showing you here. And we can see these on the surface of the placenta. Their job is to start invading. So here we have tissue sections. This is the real deal. These black cells are baby cells, and you can see that the cells are aggressively invading the mother. You don't need to be a pathologist to say this is, and if you saw this in a normal tissue, you'd say that person would be dead in a matter of days or weeks. But this is normal pregnancy, this aggressive invasion. So now let me show you what these baby cells do to the arteries. So here's a normal uterine artery. It's about 20 to 30 microns. It's a tiny, tiny structure. In pregnancy, what happens is those cells I showed you, those very invasive cells, they are able to target arteries such as this, and they totally replace the mother's cells in those arteries. So now we have baby cells lining this blood vessel. And if you look at the micron bars, which are hard to see, but these are 10 microns, these vessels become thousands of microns in diameter. And that's how we get blood to the baby. So during pregnancy, we have insertion of this novel temporary organ into the blood. And it's an unprecedented opportunity to find biomarkers. And I just wanted to show you some more pictures of this. I personally can spend hours and hours looking at these because it's just such an amazing thing to me that the placenta has figured out how to become vasculature. This is these cells come from the kind of embryological predecessor that forms your skin. And they have the plasticity to go in. This is looking inside the mother's blood vessels, to be able to go inside the blood vessel and to be able to replace the endothelial lining. There are none of the mother cells left here. These are all baby cells. Okay, so we decided to take advantage of the fact that we knew a lot about the placenta normally, how it should form normally, to start working on pregnancy complications. And the first one we worked on is one called preeclampsia. And in Greek, it comes from the Greek term eclampsis or lightning. And the reason it's called preeclampsia is that it happens so quickly. We have seen women in our clinics in the afternoon, they're totally fine. And by the evening, they're at death's door. It can happen that quickly. This condition affects about 7% of all women in the U.S. who are pregnant for the first time. So this drives prenatal care. The rule-outs now are looking for high blood pressure in women, protein in the urine, which is a sign of vascular damage, and the baby stops growing. So you can imagine that there are no tests for this, and the drives all of prenatal care. That's why women are asked to come in and get their blood pressure taken, to give urine samples, is to rule out preeclampsia. So it's a huge driver of healthcare costs for pregnant women. So I just want to go over what happens in preeclampsia. I showed all this invasion, this scary-looking invasion into the uterus. And I give this talk for high schools in the Bay Area, and it's the best abstinence talk there can be. There also, I get a bunch of people coming up to me afterwards and saying, does this really happen? Okay, so what happens in preeclampsia, we have this very aggressive invasion in preeclampsia. The placenta doesn't invade right. And so you don't get the small arteries and blood vessels that are normally bringing blood to the uterus, don't expand into these huge channels. And I just want to show you a little bit about this. When I first got involved in this, I'm not the kind of person who takes stuff in the literature for granted, and so we actually spent a huge time making tissue collections and tissue banks to ask, is this true? And this is from our own work, and this is normal pregnancy, which I've shown you a lot about, but remember those black cells in regular pregnancy? Well, in preeclampsia, it's like they hit a wall. They can't get into the uterus. So now we're seeing a pregnancy condition where there's not this right interface forming between the placenta and the maternal blood. So you can see, again, this might be a really interesting place for us to look for biomarkers. So another thing that we have done is we've taken advantage of the fact that these cells, I told you, they come from an embryological origin that's not like the vascular system. They make, we have found out, every angiogenic and even lymphangiogenic molecule that's known. So they're not leaving anything to chance. Usually your blood vessels make only subsets of these molecules. And I think these are VEGFs, which are the opposition arm is used now in cancer therapies. And these are very famous molecules in vascular biology. The placenta makes all of them, essentially, and some of them are used in preeclampsia. Molecules that we discovered are used for prognosis in preeclampsia, but they're not very good So I just want to show you a little bit. This is sections of the placenta, and we showed many years ago, and others took this forward to the clinic. The black staining is VEGF expression normally, and I think you can see in preeclampsia, here's a woman who was delivered at 32 weeks. Here's another patient at 32 weeks. Their cells have no VEGF in them. So this led to the hypothesis that preeclampsia is a disorder of angiogenic molecular production at some level. And we did this work. We found that first trimester, second trimester, third trimester, normal placenta made a certain amount of a soluble VEGF receptor. There was a lot more in preeclampsia. So we decided not to move this molecule forward, but colleagues from Harvard and the NIH did move this molecule forward. There have been some tests of it. There are some New England Journal articles, but we were not as impressed by the differences as we would like to be. So we sort of moved on. So how did we do this? Well, the other part of my lab is a mass spectrometry lab, and we do a lot of biological mass spectrometry, and in recent years we've been very involved in biomarker discovery and proteomics. So our goal here was to collect blood samples, and I'm just going to pause to say for a moment that I cannot overemphasize the importance of having the banks you need of the sample types required, in our case blood, to be able to come up with biomarkers. So we started with colleagues around the world in the early 2000s collecting first and second trimester bloods, blindly not knowing the outcome of pregnancy so that we would have the blood samples we need. And this is a huge stumbling block in most of these types of studies because we can be as visionary as we want about the proteomic and health applications. We don't, in most cases, we don't have the sample banks required to test our biomarkers. So I just want to say a word about mass spectrometry. It's my technology. I love it. And so I want to say a few words. It's gas phase chemistry. You take a sample, you put it into the gas phase, and mass spectrometers are able to weigh things very precisely. Very precisely. And part of the elegance of human biology and human structural biology is if you weigh our molecules very precisely, you can identify them. And mass spectrometers are big, complicated machines, but all they do is weigh things very precisely. And so I just thought I'd show you a little bit. I personally just absolutely love this data. Larry showed his dots, and we saw some of the genome sequencing, and I think scientists just love their data. So when we weigh things, we get the molecular mass in a peak of how much that thing weighed with some hydrogen ions tacked onto it. So from this, we can deduce what it was. So we've been using a technique that we didn't come up with, but we're some of the testers along with Applied Biosystems and Life Technologies. They came up with a molecular cassette. So here's the molecular weight of something up here, and there are mass tags we can add that allow quantification. So the mass tags fragment, so you get a reporter ion that tells you quantification, and you get identification. So now we have a reagent that we can put on a sample, and it's so far an 8-plex. And so we take our 8, in this case, blood samples from normal women and women with preeclampsia, and we put on this reporter tag that's going to tell us how much of that protein was in that sample. And it's a very slick technique, and it works extremely well. So we're going to look at our reporters. The tags vary in molecular mass by 1 Dalton. So let's see how we can use this. We just started out with an extremely small group of samples. We picked very elite cases from 6 women who had completely normal pregnancies and 6 that were had very severe and early preeclampsia. We decided to take samples from Japan because we wanted right off the bat to make a test that would be helpful to everybody and not just people here in the U.S. So our first experiments were very exciting. Here is a molecular weight measurement down here, and here are our tags. And we found right away that in patients with preeclampsia, PE versus normals, we had more of this one protein. And we were extremely excited about that until I went up to my office and found out that this, what PEDF stood for. And PEDF stands for pigment epithelium-derived factor. And PEDF is a molecule that's made in the eye. And I was like, oh my gosh, we found an eye molecule in pregnancy. This is probably not going to make all that much sense. But fortunately, it did make sense. And this is quantification. I think I'm going to skip this. So what did PEDF do? PEDF plays a huge role in angiogenesis. And I told you that I thought preeclampsia has a lot to do with angiogenesis. So let me tell you what PEDF does. In angiogenesis, we have formation of new blood vessels from pre-existing ones. And we know that in cancer, angiogenesis is a big problem. We get neoangiogenesis in response to tumors. Vasculogenesis is how you get blood vessels in the first place, the new formation of blood vessels. So we know that angiogenesis relies on molecules that stimulate this process. Here's a whole laundry list. And here are molecules that inhibit this process. So it's a very big balancing act. What PEDF does is it inhibits this process. And I particularly want to call your attention to, it's thought to be the most powerful anti-angiogenesis molecule known. And many people probably haven't even heard of it, and it's one of those molecules that got named where it was first found in the retinal pigmented epithelium of the eye, and not essentially for what it does, which is hugely anti-angiogenic, and it's in clinical trials now like Avastin for cancer treatment. And here we can see what it does in the eye. It's anti-angiogenic, but in hypoxia, which is created in diabetes, the hypoxia decreases VEGF production, and it increases PEDF in the first arm of this. But during the very, very, very last stages of the disease, the flip happens. And so you get tremendous amounts of VEGF production, no PEDF, and then you get this overgrowth of blood vessels in diabetes. So it's very dramatic what this molecule does biologically. So now we know that the placenta gets inserted into the maternal bloodstream. We know that in maternal blood and preeclampsia, we can find a lot of this PEDF anti-angiogenesis molecule. Can we tie the two together? So we hypothesized that the placenta was actually where the PEDF was coming from. So here we show This is immunofluorescence. And so when you see color, and in this case, the green color shows PEDF in the placenta, I think what you can appreciate, this is normal placenta A here on the top, and C, we can see preeclampsia, and so you can see tons more of the green. And the interesting thing about the green is not coming from the placental cells itself, it's coming from the intrinsic blood vessels of the placenta. It has its own blood vessels. And until we saw these pictures, we had always thought, well, all the biomarkers will come from those invasive cells. But here we have things coming from the blood vessels themselves. And so here are the invasive cells, and I just want to show you, we have an interesting piece of biology. We think this molecule is coming from the blood vessels itself. The invasive cells, which are here, are making the receptors, and so we think that this is crosstalk between blood vessels of the placenta and the invasion process. And we actually are able to prove this. So this is an invasion assay in which we are able to add PEDF to isolated invasive trophoblast cells in culture. This graph shows you that with PEDF we have a lot less invasion, and I told you that was the phenotype of preeclampsia. And this is a molecular fingerprint showing not only does invasion decrease, but we get the molecular phenotype of preeclampsia. For example, this is this VEGF molecule that we had discovered years ago and is used as one of the prenatal tests for preeclampsia. Here, very little of it is made by the cells. A lot is made when we add PEDF. But can this be a biomarker? So we went then to our clinics. We've had a program project in preeclampsia for many years. We collect blood samples at 16 weeks and at time of diagnosis. Our worldwide collaborators are collecting blood at 16 and 20 weeks of gestation and at time of diagnosis. And our Japanese collaborators are doing the same. So we got together these samples, and these are the controls. So this is a control blood protein. This is all immunoblotting, where there's not yet an ELISA that works for this molecule, or the usual kind of antibody test in solution. So this is testing an immobilized antibody on an immobilized sample. And I just want to draw your attention to the kinds of numbers. Here are time of disease from 3 different banks. And if you look at the number of cases and controls, and here is 20 weeks. And we have 40 samples taken at 20 weeks before a woman knows she's going to get preeclampsia in 20 weeks after. So you can imagine, to get 40 of these women, there's had to be many thousands of women screened. So I just wanted to applaud our clinical collaborators and say that it really takes everybody. So I think you can see the preeclampsia patients are on the left. We see very strong bands in these patients, and in general, the PEDF levels on the controls on the right are not as high, and the ratios are shown for you out here. So I don't want to make too much of this. This is not absolutely a perfect test, but as a relatively small-time academic lab, we have been able to advance the concept that the placenta can be an important source of biomarkers that actually could have clinical utility in pregnancy. And so you can see some of the odds ratios. They're pretty impressive for 20 weeks of pregnancy. I don't know if these will actually hold up when we have a bona fide test. We've been holding off on publishing this because we don't want to be wrong, and we want to make a more standard test, which is this soluble antibody antigen test. Oops, sorry. So conclusions, I just wanted to say we're very high. There are some people who are not very high on using mass spectrometry proteomic technologies, which is, I said this for Larry so he could raise his hand, are not very high on this, and I think you have to pick your problem. And I think for somebody like us who's looking at products of a new defective organ, it's a good technology. We think that we found an anti-angiogenic molecule with biology and biomarker capabilities. But I wanted to get to the bigger picture, because if we talk about personalized medicine, I think one thing that people are coming to a consensus on is that your adult health is programmed in the womb. So until now, our attitude about pregnancy has been let nature take its course. We'll worry about the baby's health once it's born. And we can no longer do that because we know that the roots of human health, and I'm not going to go into this, but it's called the developmental origins of adult disease. If you have a rough time in the womb for whatever reason, preterm labor, early birth, infection, starvation, if your mother had limited access to food, you have an enormously higher risk of cardiovascular disease and diabetes. And so this really behooves us to start thinking about personal medicine beginning in the womb. And we also have a wonderful chance. We have this new organ. This is not an organ that's been your body has ever seen before. It makes all these novel proteins. It gets inserted into the mother's circulation. Let's use this as a way to monitor prenatal health. And I just wanted to show you, these are all the authors of our study. You can see all the people who have collaborated with us from all around the world. It's taken a huge group of people, clinicians, basic scientists. And thank you for your attention. Yes, so that's one of the things you can see. Well, our lab is not doing just blinded biomarker discovery. We want to put it in the context of biology, and that's exactly what we would want to do. If we think this molecule is rising in the circulation long before the symptoms appear, then we have a great target to go after. And I myself would choose an antibody therapy right now, but maybe there are better ways. Yeah.
Just for a kind of a quick follow-up, it was kind of implied that it's kind of a dangerous procedure, to go in and do the sampling whilst the woman is pregnant. So there needs to be some sort of medical necessity for that.
Pardon me, could you?
As far as doing a sampling of the placenta to try to determine certain biofactors affecting the outcome of that pregnancy, how might that become more? In other words, more benign development in the future, so it's more available. Because you hear in certain circles with genetics that there's people wanting to choose outcomes of their children's in certain types, and so that's kind of beyond the scope of what we're talking about here. But just basically a non-invasive means of obtaining information for the overall outcome of the pregnancy. Is there a certain development or a certain area or track that you're going in that direction?
So maybe this wasn't clear, but the test we did was non-invasive. So we were taking blood samples from women, and our goal is to come up with non-invasive methods. And I thank you for asking that question, because I didn't mention aneuploidy, but we're doing equally large programs in aneuploidy, because we believe that if you have an organ with an extra chromosome, that you should be able to predict that from a change in the repertoire of the soluble factors it makes. So what we're going for is entirely non-invasive.
I have a question.
Oh, yeah.
No, this is...
Bring it on.
No. It's actually a follow-up to Tom's question. What is the conventional way to treat preeclampsia, or is there nothing that anyone does when a woman is in trouble?
Yeah, so the first time you know that a woman has preeclampsia is when you meet the diagnostic criteria for proteinuria and blood pressure. Those are the two things that get you diagnosed in the U.S. today. So the first thing that happens is they bring women in, and you get the standard, which is hydration. And then if the blood pressure doesn't go down, they just watch to see if bed rest will bring the blood pressure down. In true preeclamptics, it doesn't. It just keeps going. There's almost no upper limit. Then they try pressors, but conventional pressors don't work in these situations either. So the cure for this disease, and it is done every day of the week, is to deliver the baby, no matter what the age, because the women will die of the vascular component of the disease. They'll go into frank eclampsia, liver failure, other organ failure, and seizures. So taking out the placenta is the only known cure.
I just want to add to that the problem is that, and actually as a speaker of modern Greek, you know, when you have someone who's preeclamptic, it's really catastrophic, because the rate of progression can be profound and terrifying. And I work in small community hospitals, so we have to life flight people out. Because conventional ambulance transport really often isn't fast enough. And the biggest problem with preeclampsia that we worry about is stroke, because people seize, and we can't really stop it. So, you know, you can have a baby at a mother and baby couplet at 22 weeks who you're going to deliver, and, you know, the chances for that baby are really nil, and also for that mother. So my question is, in terms of non-invasive technologies, what is it? Are you thinking sort of an immunization approach here if we detect people, or what do you have in mind? I'm curious and desperate, actually.
Yeah, what we envision is being able to come up with a suite of tests that will be able to detect first women in, hopefully, even in the first trimester. This is 20 weeks, second trimester, that we'll be able to detect women, even in first trimester, who will be destined for preeclampsia. And we hope and envision that we can get a marker of such tests. This is only the first thing we found. We've tried once in our biomarker discovery. So we think that there must be underlying this a huge span of molecules that could be used for detection. And then we hope for therapy if we can get a snapshot, or not a snapshot, but a good understanding of the abnormal angiogenic communication between the placenta and the mother. Then we can use therapeutic means like antibodies to bring these into the normal range. And so we hope that diagnosis and prognosis could be coupled with therapy, because there's nothing out there now.